Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today I am going to present the topic plant growth analysis. So let's proceed. Growth is a vital process that reflects the permanent and irreversible changes in plant parts with respect to its size. Like plants are growing from germinating stage to harvesting stage, and its changing form from vegetative to reproductive stage, then weight, increase in dry matter weight and volume like grain yields or biological yields. Growth is restricted only to living cells, and it is accomplished by metabolic processes which involving the synthesis of macromolecules like nucleic acid, proteins, lipids, polysaccharides, starch, etc. The term growth is used to denote an increase in size by cell division and cell enlargement, together with the synthesis of new cellulose material and the organization of cellular organelles. Before studying in details about sigmoid growth curve, first we should have understanding of growth and development. Growth is defined as a permanent and irreversible increase in size and volume of plant accompanied by increase in dry weight, while development refers to the qualitative changes in plant parts which reflects to the reproductive growth like formation of flowers, fruits, falling of leaves, etc. In plants, the growth is of two types. First is vegetative growth and second reproductive growth. Vegetative growth occurs from germination stage to floral primordial stage, and within this period the main events which are taking place in plant life cycle are germination, seedling emergence, leaf and stem growth. Under reproductive growth, it occurs from initiation of floral primordia development to the completion of seed formation, and there are various events which are taking place within reproductive growth cycles like primordia initiation, flower emergence, anthesis, flowering, pollination, fertilization, seed development and maturation. So this reproductive growth is covering the grain yield part. Here to study the nature of plant growth. The plant growth is of two types. First one is determinate growth and second indeterminate growth. In determinate growth, the reproductive growth takes place after completion of vegetative growth. It can be defined as plant growth in which it grows to certain size and then stop growing due to presence of terminal inflorescence. After their growth is completed, they eventually senescence and die after reproductive phase. Second is indeterminate growth, in which the reproductive growth runs simultaneously along with vegetative growth of the plant. It can be defined as the plant growth in which the main stem continues to elongate indefinitely without being limited by a terminal inflorescence or other reproductive structure. Measurement of growth. Growth can be measured by a variety of parameters like fresh weight, dry weight, length of the plants and area. The measurement of growth is possible in terms of increase in weight, increase in volume, increase in area and increase in size. And the methods which are adopted for the measurement of plant growth is using the scales, second by using horizontal microscope, and third using oxynometers. The common and simplest method for the measurement of growth can be direct method by which the growth is measured by a scale at regular intervals from beginning to end. In other methods, the horizontal microscope and oxynometers are used for various parameters as described. Types of growth measurements. These are based on various parameters. Like first one is linear measurement. Generally growth is measured in terms of increase in length of root and shoot. A stem length is taken from soil surface to the tip of uppermost node. There are some limitations or constraints like variation in seedling growth. In dark, a stem elongates more but remains thin, whereas in light, seedling is short and thick. Second measurement is dry weight measurement, where the plant material is dried at 80 degrees centigrade temperature for about 2 or 3 days till getting constant weight. In this measurement also there are some constraints like dry matter may increase due to deposition of food material like a starch, or may decrease also due to oxidation of food material as a result of excess heating. So there are some chances of error to get the actual measurement. Third is fresh weight measurement, where weight of freshly harvested plant is taken as fresh weight. Here also there are some constraints like plant has to be removed while measuring fresh weight. It depends on water content of tissue, which keeps on changing due to variation in rate of absorption and transpiration. Another measurement is leaf area measurement. The measurement of leaf area is also taken as an index of growth, and some limitations like accurate determination is difficult without having sophisticated instrument for the measurement. Next is volume measurement, where the volume of plant part or plant can be measured by water displacement method. And here the constraint, it is not accurate method due to difference in compactness of tissues of various plant parts. Now to study the growth analysis concept. Growth analysis is a mathematical expression of environmental effects on the growth and development of crop plants. This is a useful tool in studying the complex interactions between the plant growth and the environment. The basic principle behind this concept of analysis is the estimation of crop growth at various stages and finally reasoning for yield variations. This would give an insight not only on the performance of a particular genotype but also on the impact of superimposed agronomic practices on the crop at any particular stage of growth as well as on the final yield. The problem of accounting for variation in yield in terms of growth and development of the crop is very complex because of the effect of external environment on the plant physiological processes, the interrelation between different physiological processes, and the dependence of the above two on internal factors determined by the genetic constitution of the plants. Now to go through objectives of growth analysis. First objective, to identify a spatial and temporal integration of all plant process. Second objective, to know the rate of dry matter accumulation which varies across the plant life cycle. Third, to quantify the effects of environmental influence or to analyze the genotypic differences between crop cultivars. Fourth objective, to learn what are the most relevant methodologies to measure the daily performance of crop canopy. And fifth one, to know why does plant to plant variability makes the measurement of plant productivity so difficult. For growth analysis, various parameters are used, like first one, crop growth rate CGR, second is relative growth rate RGR, third is net assimilation rate NAR, fourth is leaf area index LAI, fifth is leaf area duration LAD, and sixth leaf area ratio LAR, about which we will discuss in details. First parameter is crop growth rate CGR. Crop growth rate is the gain in dry matter production on a unit of land in a unit of time. And this concept was first described by Watson in 1952. The crop growth rate simply indicates the change in dry matter accumulation over a period of time. Crop growth rate is a measure of the increase in size, mass, or number of crops over a period of time. This expression can be used without any assumption, without the form of growth curve. The formula can be used to compare treatments between and within experiments. They can even be used when it is possible to make only two harvests. The formula of CGR, crop growth rate, is defined as W2 minus W1 divided by T2 minus T1, where W1 is equal to dry weight per unit area at first sampling T1, W2 is equal to dry weight per unit area at T2, second sampling. T1 refers to the first sampling and T2 refers to the second sampling. And the unit of CGR is gram per square meter per unit time. It may be day. Second parameter is relative growth rate RGR. The concept was first given by Blackman in 1919. The relative growth rate expresses the dry weight increase in time interval in relation to the initial weight. Relative growth rate is the slope of a curve that represents logarithmic growth over a period of time. An exponential growth rate is not sustainable over time. The curve typically flattens out, representing saturation in a growth at a certain point of time. In practical situations, the mean relative growth rate is calculated by the formula RGR is equal to log E W2 minus log E W1 divided by T2 minus T1, where W1 is equal to dry weight per unit area at T1 first sampling, W2 is equal to dry weight per unit area at T2 second sampling. T1 refers to the first sampling time and T2 second sampling duration. And the unit of relative growth rate is represented by gram per day. In plant physiology, relative growth rate is a measure used to quantify the speed of plant growth. It is measured the mass increase per above ground biomass per day. It is considered to be the most widely used way of estimating plant growth. Third growth analysis parameter is net assimilation rate NAR. Net assimilation rate is the net gain of assimilate per unit of leaf area and time. NAR expresses plant's capacity to increase dry weight in terms of the area. The concept was first given by Gregory in 1918, and it directly indicates the rate of net photosynthesis and expressed as gram of dry matter produced per day per square meter of leaf area in a day of its assimilated surface. For calculating net assimilation rate, the formula used W2 minus W1 multiplied by log E LA2 minus log E LA1 divided by T2 minus T1 into multiplied by LA2 minus LA1, where W1 is equal to dry weight per unit area at T1, W2 is equal to dry matter per unit area at T2, LA1 is equal to leaf surface area at T1, and LA2 is equal to leaf area at T2. where the T1 and T2 represents first sampling and second sampling duration, and the unit of NAR is gram per square meter per week or gram per square meter per day. Since net assimilation rate gives no direct indication of respiratory losses, this index doesn't necessarily serve as a measure of inherent photosynthetic capacities. The third limitation is of importance to the agronomist. While net assimilation rate indicates plant efficiency in producing dry matter, economic yield is subjected to additional controls and is not necessarily related to photosynthetic efficiency. Fourth parameter is absolute growth rate, AGR. This concept was first given by West et al. In 1925, and this indicates the rate of increase of total dry weight per plant. Absolute growth rate aims at what rate the crop is growing. Like example, whether the crop is growing at faster rate or slower rate than normal. The simplest index of the plant growth, a rate of change in size, an increment in size per unit time. Most commonly applied to total dry weight or total leaf area per plant. To calculate absolute growth rate, the formula used is W2 minus W1 divided by T2 minus T1, where W1 is equal to dry weight of plant at time 1 sampling in gram, W2 is equal to dry weight of plant at second time of sampling T2, T1 is equal to time 1 in days of sampling, T2 is equal to sampling time in days, and the unit of absolute growth rate is gram dry matter per day. Fifth parameter is leaf area index, LAI. Leaf area index is the ratio of leaf area to the area of ground cover. Means it is the leaf area on surface only divided by the land occupied by the plants, and leaf area index having no unit, it is a unitless figure. The concept of leaf area index was given by Watson in 1947, and leaf area is important for photosynthesis, and its estimation is related to both assimilating area and growth. For crop production, leaf area per unit land is more important area of individual plant. Leaf area typically increases after crop emergence to a maximum and then decline. For maximum production of dry matter of most crops, leaf area index of 3 to 5 is usually necessary, and values required for maximum production increases with the level of solar radiation. And the formula of measuring leaf area index is equal to LA divided by GA, where LA is equal to leaf area and GA is equal to ground area. The leaf area index at which the canopy first reaches maximum crop growth rate is called critical leaf area index. The leaf area index with 95% solar radiation interception has been adopted as the critical leaf area index by most crop physiologists. The leaf area index at maximum crop growth rate is called optimum leaf area index because the crop growth rate decreases as the leaf area index increases beyond the optimum level. Here to study optimum leaf area index. Leaf area index increases slowly in early stage of crop growth and rapidly after seedling stage. As leaf area index increases, light interception is more resulting in dry matter production. However, at high leaf area index, mutual shedding of leaves occur, and as shaded leaves respire more than unshaded leaves, they contribute less to the dry matter production. There is therefore an optimum leaf area index for maximum dry matter production, which is reached when the largest number of leaves receive just sufficient sunlight for photosynthesis to balance respiration. Below optimum leaf area index, light is not being fully intercepted, and above optimum leaf area index, the leaf area is not being fully utilized at maximum efficiency. At higher than optimum leaf area index, lower leaves become parasitic even under conditions of full light intensity. Optimum leaf area index differs with crop and their leaf orientations. Optimum leaf area index is between 3 to 4 for crops with horizontally oriented leaves, and optimum leaf area index is in between 6 to 9 for crops with upright leaves. Sixth parameter is leaf area duration, LAD. Leaf area duration expresses the magnitude and persistence of leaf area or leafiness during the period of crop growth. Leaf area duration reflects the extent or seasonal integral of light interception. Leaf area duration is measured with the formula LA2 plus LA1 multiplied by T2 minus T1 divided by 2, where LA1 and LA2 are leaf areas at time T1 and T2 respectively, and its unit is per square centimeter per day. Yield of dry matter is a function of leaf area, net assimilation and duration of leaf area. Leaf area duration of a crop is a measure of its ability to produce leaf area on unit area of land through its life. If leaf area index is plotted against time, it produces a function that indicates assimilatory capacity of crop during the period. The leaf area index declines rapidly in some crops, which may restrict growth management practices, which can prolong the duration of leaf surface in an active state, may improve the yield. Leaf area duration provides a means for comparing treatments on the basis of their persistence. It is usually determined by measuring the area beneath leaf growth curve for selected parts of the season. Seventh parameter is leaf area ratio, LAR. Leaf area ratio is the ratio of the total leaf area to the whole plant dry weight and is a further measure of the efficiency of leaf surface in producing dry matter. It can be calculated by the formula LA1 divided by W1 plus LA2 divided by W2 and divided by 2, where LA1 and LA2 are the leaf area at dry weight W1 and W2 respectively, and the unit of leaf area ratio is per square meter per gram. Here to study the advantages of growth analysis. We can study the growth of the population or plant community in a precise way with the availability of raw data on different growth parameters. The studies involve in assessment of the primary production of vegetation in the field at the ecosystem level of organization. The primary production plays an important role in the energetic of the whole ecosystem. The studies also provide precise information on the nature of the plant and environment interaction in a particular habitat. The study of growth analysis also provides accurate measurement of whole plant growth performance in an integrated manner at different intervals of time. Along with the advantages, there are some drawbacks or limitations also of growth analysis study. Like in classical growth analysis, sampling for primary values consist of harvesting representative sets of plants or plots, and it is impossible to follow the same plants or plots throughout a whole experiment because we are harvesting the plants. Here to go through factors which are influencing plant growth. Growth is affected by all factors that affect the activity of protoplasm. Both physiological and environmental factors such as water, minerals, photosynthesis activity, respiration, climate, and edaphic factors significantly influence the growth. In general, the factors which are influencing growth are categorized in two types. First one is internal factors and second external factors. As internal factors are concerned, there are growth hormones and their availability which are controlling or affecting growth. Second is resistance to climatic, edaphic and biological stresses are another factor. Third is photosynthetic rate and respiration of the plants depending upon the climate situations and availability of the nutrients. Fourth is assimilate, partitioning and nitrogen content. Fifth is chlorophyll content and other pigments related to the metabolic activities, photosynthetic activity in the plant. Sixth is source-sink relationship and enzyme activities. So these are the internal constituents which are in plant which are reflecting the growth. Second factor is external factors, which is consisting of light, temperature, oxygen, carbon dioxide, water, nutrient and soil as major factors. First external factor is light. Light has direct effect on photosynthesis and transpiration. Light in terms of intensity, quality and periodicity influence the growth very much. First one is light intensity. A weak light promotes shortening of internodes and affects expansion of leaves. Very weak light reduces the rate of overall growth and also photosynthetic activity due to poor development of chlorophyll and higher rate of water loss from the plant. Now light quality. The different wavelengths of light have different responses to growth. In blue-violet radiation, the internodal growth is pronounced, while green color light promotes the expansion of leaves as compared to complete spectrum of visible light. The red light favors the growth, while infrared and ultraviolet light is detrimental to the growth. Third light factor is light duration. There is remarkable effect on the duration of light on the growth. The induction and suppression of flowering depends on duration of light. Second external factor is temperature. The plants have different temperature requirement based on the region where they are grown. In general, best growth takes place between 28 to 33 degrees centigrades and it varies from temperate to tropical conditions. The optimum temperature requirement is essential for seed germination, growth, metabolic activities, flowering and high yielding. Third factor is oxygen. The growth of the plant is directly proportional to the amount of oxygen which is essential for respiration, during which the food materials are oxidized to release energy. Fourth external factor is carbon dioxide, which is one of the major factors that influence the photosynthesis. The rate of photosynthesis increases as the availability of carbon dioxide increases, while other factors are not limiting. Fifth factor is water. Water is essential for growth because it is controlling the uptake of nutrients, translocation of nutrients and food material through the plant cycle system. Water is regulating the transpiration and various physiological processes like photosynthesis, respiration and enzymatic activities are required water. Sixth factor is nutrient and food materials. The rate of growth is directly proportional to the availability of nutrients and food materials. The shortage of food supply affects the growth as it provides the growth material to the growing region and also it provides the potential energy to the growing region. Seventh factor is soil, which provides the anchorage for plant growth and development, serves as a medium for water and air circulations, and soil acts as a reservoir for water and nutrients and provides a space for beneficial microorganisms. Now we will focus on growth curves during plant life cycle. Growth curve is a graph obtained by plotting the growth rate of a plant against time factor. The growth rate of a cell or a plant organ or a whole plant or whole life cycle of plant is measured in terms of length, size, area, volume or weight. And it is found that different growth phases results in S-shaped curve, which is known as sigmoid curve. In initial stages, during the phase of cell formation, the growth rate increases slowly, while it increases rapidly during the phase of cell elongation or cell enlargement, and again slows down during the phase of cell maturation. These growth curves are divided in three types. First one is sigmoid curve, second is polynomial curve, and third is asymptotic curve. As sigmoid curves is concerned, there is typical growth pattern of annual plants, and under a sigmoid curve there are three phases. First one is lag phase of growth, second is log phase of growth, and third is senescence or steady or stationary phase. First we would have introduction about sigmoid growth curve. The rate of growth of a plant depending upon the various factors, and it is not always the same. Sometimes the growth rate is very slow and sometimes there is rapid nature. If we plot the increase in cell numbers regarding growth rate against time, a typical S-shaped curve is obtained. This is called growth curve or sigmoid growth curve. Now we will study the phases in sigmoid growth curve. First phase is lag phase of growth. In this period, the growth rate is quite slow because it is the initial stage of growth. Second phase is log phase of growth, or also known as rapid phase. During this period, the growth rate is maximum and reaches the top because at this stage the cell division and physiological process are quite fast. So in this period there will be rapid growth of plants. Third phase is senescence phase or steady phase, where in this period the growth is almost complete and it's become static. So the growth rate becomes zero and the crop moves towards the maturity and harvesting stage. Second type of growth curve is asymptotic growth curve. When yield is the product of vegetative crop growth, the density yield relationship is asymptotic. In an asymptotic relationship with increase in density, yield rises to a maximum and then relatively constant at high densities. Further increase in plant density above the maximum doesn't increase the yield. This partially explains why recommended seed rates for fodders are higher than that for grain. The curve for biological yield can be defined by the expression for a rectangular hyperbole. Third type of growth curve is polynomial growth curve, and it is also known as parabolic growth curve. The parabolic response curve is typically a flat-topped one with decrease in grain yield on both sides of an optimum. The maximum plant yield is reduced by the increasing competition resulting from greater plant density. In consequence, it may be termed as competition function. So this presentation was all about plant growth analysis. Hope this will be useful to all of you. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Shrivastav. Having request, please visit the channel, subscribe it, and provide your kind and valuable feedback. Thank you.